animal health since their discovery. And the first natural antibiotic that was discovered by Alexander Fleming was in 1928, after which there was an explosion of more discoveries after that. And from 1950s up to now, more than 30 similar or related antibiotics that are in use both in human as well as in agriculture for disease prevention as well as treatment of various diseases. And you find in some countries, these antibiotics are used more in agriculture than in human. That is depending on the human animal population in those countries. Like for example, in Chile, you find 10-fold of the quinolones are used more in agriculture than in the human medicine. And then you find like in Africa and North America, most of these antibiotics are used in agriculture in low doses for the promotion of growth in these animals, especially in Africa, you find in chickens as well as in pigs. And this practice was also in EU. However, it was discouraged and it's no longer being practiced. And I'm hoping Africa and North America will also follow suit, because as Prof has told you, this use of these antibiotics end up creating the superbugs, which are resistant to multiple antibiotics. And if we are not careful, we may reach an era where antibiotics are no longer useful for both human and animal purposes. Now, the misuse of these antimicrobials, as you've been informed, usually creates the superbugs that are no longer susceptible to these available therapies. And man is not the only culprit of AMR or development of AMR, but you find also these microorganisms, as Prof has told you, are able to develop innate resistance, where these microorganisms do not respond to these antibiotics. And for example, in Yersinia pestis, resistance against doxycycline, which is a drug of choice in the treatment of Yersinia pestis, has been seen to be resistant. And when they do studies, they have realized that it acquired natural resistance towards this doxycycline. Now, whenever these bacteria or these microorganisms develop this resistance, they can exchange this information among the related bacteria or even among different species or different genuses of this bacteria. And there are two main mechanisms that they will exchange this information. This is either through vertical gene transfer or through horizontal gene transfer. And vertical gene transfer is basically the exchange of this information or the resistance information coding for resistance towards the different antibiotics from the parent to the offspring. And this one usually happens naturally through either sexual reproduction or asexual reproduction. And as you know, with bacteria, it usually has asexual reproduction by binary fusion, where one parent cell splits into two to bring up two daughter cells. And the information that was being coded in the parent cell end up being passed to the offsprings. This is a bit different with the horizontal gene transfer, whereby the information is not being passed to the offspring, but the resistance, the information according for this resistant is being passed from one microbe to another, and sometimes this may not be even related organisms. And there are different, horizontal gene transfer is also known as the lateral gene transfer, And it is very common among bacteria as well as the archaea. Unlike with eukaryotes, we hardly get horizontal gene transfer. Now, with horizontal gene transfer, it can occur in three main methods, either through conjugation, transduction, or transformation. And most of these methods are dependent on mobile elements, especially the plasmids that will have the information that is coding for this drug resistance. Now, if we look closely at each of these methods, Underberg and his colleague Nkatha in bacteria, when they come in close contact, they are able to exchange information. And basically for that to happen, there has to be a pili involved. And apart from a pili, which normally bridges the donor cell to the recipient cell, and then information is passed, they are also dependent on plasmids. These are extra chromosomal DNA that will be encoding for that information, which we are saying is antibiotic resistance. Now, this is an illustration of conjugation, whereby the donor cell has to attach itself to the recipient. And once this information is passed, you find that the recipient is the one that doesn't have the information coding for that resistance. But once the sex pili attaches itself to this recipient cell, the information on the donor cell is exchanged to the recipient cell. And finally, now the donor cell, which didn't have information coding for the drug resistance, will start exhibiting that resistance. And then in case the donor or the donor cell that has gotten that information, the information can be integrated into the host chromosome, and this creates what we call a high frequency recombinant cell in nature. Now, the second is the transduction, and transduction was also first described by still Joshua Landerberg, who also described conjugation, but he was accompanied by Norton Zinder, and that was 1966. And they were trying to study the phages, and they realized that bacteriophages can help in the transfer of information from a donor cell to a recipient cell. And this involves both the lytic as well as the lysogenic phages, bacteriophages. And unlike conjugation, which happens even in distantly related bacteria, most of the time transduction happens in closely related strains. And we have different types of transduction. We have the generalized transduction, where the bacteriophage randomly picks up any portion of the host DNA, and then it is transferred into the recipient cell, But this one is rare. It occurs in 1 out of around 11,000 phages, and it has been described in the E. coli, especially using the P1 phages. This is a bit different from the specialized transduction, whereby the bacteriophage picks up only the specific portion of the host DNA. And you find this one is very specific, and that portion is the one that is transferred into the donor, into the recipient cell. And this has been seen in the E. coli transduction by the Grabda phages. And then finally is a bit more transfer of genes at higher frequencies than the generalized as well as the specialized transduction. And this one has only been described in the Staphylococcus aureus. Then finally, we have the transformation, which was first described by Frederick Griffith in 1923. He was trying to study strep pneumoniae and how strep pneumoniae can spread the virulence of the strep pneumoniae. However, the principle was finally described by Avery, Collin, and MacLean in 1944. And they realized that basically bacteria can pick up naked DNA from the environment, and then the recipient cells, usually now when they pick up this naked DNA, they start exhibiting the information that they have picked up. And now remember, we are talking about drug resistance, and therefore they start exhibiting resistance towards the antibiotic per se. Now, you find also with transformation, this one usually occurs among closely related species or strains. And it has been seen that transformation in the lab can be done. And in the 1980s, electroporation, whereby you can treat these bacteria using either by use of chemicals and shock, and you create pores within the recipient bacteria, allowing the bacteria to absorb the naked DNA and therefore exhibiting the resistance. And you find this one is more of an in vitro, not in vivo transformation. And it is not clear if this kind of cells can end up also being transformed under natural conditions. Now, with these main mechanisms, either the vertical or horizontal gene transfer, you find the microorganisms can have their antibiotic resistant genes being now passed from one bacteria to another. These bacteria can be in any of the environments, such as in water, in soil, in food that we eat. And once these bacteria have acquired this information, they'll start exhibiting the information. For example, if the donor was exhibiting resistance towards penicillin, then automatically the recipient start also exhibiting the resistance towards that same penicillin. Now, how do these, once these microorganisms have obtained these ARGs or the antibiotic resistant genes, they can be spread across the world in different ecosystems, in water, in food, in animals. And you find, for example, the bacteria that is resistant towards these drugs may first develop maybe in the environment, and then it is passed to animals or man, or vice versa. For example, vancomycin-resistant enterococci first was found to have developed in farms in North America, whereby there was a lot of usage of avopacin, and this avopacin was being used as an animal growth promoter. It is closely related to vancomycin, and therefore This resistant towards avopacin was passed to man, and even with the usage of vancomycin, the vancomycin no longer was exhibiting any susceptibility. This is different, whereby you find the bacterial resistance can develop in man and then get transferred to animals. For example, the methicillin-resistant Staph aureus, the ST398 strain, was first seen in man, and then it went into pigs, where it picked up the MEK-A gene, and then it came back to humans, from pigs back to humans. So this resistance can be from man to animals, or from animals to man, and vice versa. Now, also, even from the food that we consume, for example, the multi-drug resistant Salmonella strain, specifically the DT104 strain, was found in humans at first, and the animal infections which originated from the aquaculture farms in the Far East Africa, and this MDR Salmonella DT104 had the fluorophenical gene as well as the tetracycline gene that basically codes for the multi-drug resistance in Vibrio. And it was finally found that from Far East in animals, it was isolated in US as well as in Africa. Now, this is also similar to, like, for example, the Listeria monocytogenes and the Yersinia pestis that normally carry resistant genes. If you come in, for example, Listeria monocytogenes can be acquired in products, then the resistance can be transferred to the normal microbiome, PIT, and this normal flora can start exhibiting the same resistance that was being in the Listeria monocytogenes or even Yersinia. Now, in summary, we can say that the use of antibiotics normally selects for bacterial resistance, which can be transferred from one microorganism to another, either through the horizontal or vertical gene transfer. And once these microorganisms acquires this resistance, it can be passed from in our different ecosystems in the entire world. And basically, any practices in any one of the country or any region impacts greatly on the resistance in other countries. For example, you find if the usage of these drugs or growth promoters in Africa, the bacteria that gains resistance in Africa will end up crossing in America or in Asia, and you will not say that the problem is only in Africa, because eventually it will cross to other parts of the world. And any changes in the biotic usage needs to be done around the world. We should see that this is a problem not only for you and me, but it is for the entire. They end up reaching what we call a post-antibiotic era, where most of the to be useful in the treatment or in prevention of infectious diseases. Otherwise, thank you very much. I may take any questions.
Thank you so much, Lisa. That's a great session, great presentation. Really appreciate that. We're going to the next session, that is the QA part. And just would like to request anyone with a question, you can raise up your hand, you can post your question on the chat box. But we can begin with a few questions that we had earlier on. I don't know if these were answered, but I think they were not. But yeah, we can take off. And these two questions, I think they were directed to Professor Joel Bazira. So the first question is from Jeffrey. Jeffrey is asking, you're focusing more on US ICUs. Have you looked at those cases close to home? And yeah, maybe Professor, you can answer that question so that we can proceed. Over to you, Prof.
Yeah, that question was answered.
Okay, okay.
Unfortunately, you are offline, but we are looking at ICUs in Uganda, and specifically, we have some data on what we call the high risk MDR units.
Oh, sure. Thank you so much, Prof.
We have some data on that. There was somebody who asked about bacteriophages and making use of bacteriophages. That has been a proposal, but it has not really been brought into clinical practice. I mean, in theory, yes, you can use antibiotics. I mean, you can use bacteriophages to kill off bacteria, but it's still in research. Nothing yet has come on table for the clinician.
Okay, thank you, Prof. Okay. So anyone else? I'm not too sure if we have any other question unanswered, because yeah, I've been experiencing some technical issues here, but I think yeah, that was the only question that I missed. Let me hope so. Next question, please. If you have any question, the participants kindly just send your question. You can send directly in the chat box, or maybe you can even raise your hand. Yes. So we have one question from Basil Okello. He's asking, please clarify for the role of biofilms in MR. I think this goes to Dr. Lisa. Maybe Lisa, you can kindly pick up this question.
Yes, Jimmy?
So yeah, we have one question from Basil Okello. He's asking to clarification on the role of biofilms in MR.
Biofilms, these are bacteria that tend to thrive more on surfaces, and that's why we are saying even in the environment, if they get in contact, especially through conjugation, if they get information from the bacteria that is surrounding them, that as any information that is coding for drug resistance or even multi-drug resistance, then they can acquire that information and they can pass to other bacteria around them, whether they are similar species, they are similar strains, they will pass that information.
Thank you so much, Lisa. Abubakar, over to you.
Yes, thank you very much. Let me just add on the issue of biofilms. Actually, the mechanism of action, biofilms are polysaccharides that surround these organisms, and the resistance occurs when there's this colonization, the coverage of the various devices, catheters, intravenous catheters, and all those things. So this mechanism of the resistance in terms of these biofilms is based on the mechanical so in which the antibiotics cannot penetrate the organism and destroy the organism. unlike the others that is specifically and strictly genetic. So it's mostly the mechanical issues like Staphylococcus epidermidis. They tend to get a lot of buff, especially capsulated organisms. I think that is one of the addition to the clarification question. Thank you very much.
Thank you so much, Abubakar. That was so helpful. And thank you for clarifying that and for answering more on that. Thank you so much. So, Martin, please, if you had a question before, I think I missed that. You can kindly repeat, or you can resend your question, please. I can't retrace it. Martin, if you can hear me, please, you can raise your hand.
Hello, can you hear me?
Yeah, I can hear you. Go on, please.
Yeah, the question is prescribing antibiotics based on empirical evidence a good practice in view of AMR prevention and stewardship? It's actually on the chat. I think it's directed to Prof.
Okay, thank you, Prof.
Yeah, so you're saying prescribing antibiotics empirically?
Yes, Prof.
That simply means prescribing antibiotics without evidence.
Yes,
if I understand.
Contrary to what you said.
Of course, that's dangerous.
Yes.
It's one of the things that is the practices that is fueling resistance, because you're prescribing a drug without evidence on whether it works for the right cause at the right duration. So you don't have actually a diagnosis. So you're just thinking this may be the diagnosis. So that's one of the challenges that we have. That's why we are calling for antimicrobial stewardship, where you prescribe a drug for the right cause, for the right duration, using the right dosage formulation and the right route.
All right. Thank you, Prof.
Okay, thank you, Prof. And do you have any question? I think, yeah. I don't think we have any more question from the audience. But in case there is, yeah, my email is always open. You can always send me a question, or maybe even my telephone number. I think most of my people also have my telephone number. Those who are in the WhatsApp group, yeah, you can kindly continue. We can continue the discussion at that point. And also, Dr. Lisa, yeah, I think most of the people are asking for your email address, so kindly, if you can share it in the chat box, or maybe you can also allow me, I can send to those who have requested for it, because yeah, some of them have reached out, so maybe you can also share it to the requested participant. Thank you so much. Anyway, I think, yeah, the session has been so much educative, very intriguing, and I really hope that we have learned a lot. And yeah, this is just the beginning. Yeah, we are going to hold more sessions coming next month. That's on September. Yeah, we're going to have another session, and we'll be glad to have a very good audience like here today. I'm so much happy to have this very huge audience today. I really appreciate that, and I think our appreciation goes to those who have attended the session, and also hope that we are going to learn more on this, actually the fight against antimicrobial resistance. We have seen an AMR can happen anywhere, and not only in Africa, it can travel all over the world. So maybe in America, Asia, Europe, and anywhere. So I think if we become active champions against antimicrobial resistance, I think we can also play a role as the youth of the young people in mitigating the AMR. I know we have a good number of professionals in the house, and really appreciate always your support in guiding us and mentoring us and providing the support that we always request as the young people fight against antimicrobial resistance. Thank you so much. So I think without taking much time, we are going to end our session today. So I just would like to give Dr. Lisa Nkatha to say final remarks before we end of the session. Over to you, Dr. Lisa.
Oh, thank you, Jimmy. I thank everyone for attending this session. We are going to be having more sessions. We are going to do more collaboration with SARS. As a member of the American Society of Microbiology, I encourage the SARS members who are microbiologists or scientists to join also the American Society of Microbiology. Jimmy, I'll be sharing the contacts. I think on the flyer, we had put the contacts of ASM, but they can also contact me. They can inbox me, whereby I can show them how to join the ASM. There are very many benefits of becoming an ASM member, especially there is the student part. Let students not feel, oh, me as a student, how will I benefit from ASM? There are a lot of benefits. There is a lot of networking that you can benefit from. So apart from SARS, also we are going to have workshops. We'll be calling upon SARS, whereby we'll have workshops moving forward. And I look forward to working with SARS. Thank you so much. There are presentations. I'm going to share with you my presentations so that you can be able to share with all the students or those who have attended the webinar. Thank you so much, Jimmy.
Sure. Thank you so much, Lisa. And I really appreciate that. This is the second time having Dr. Lisa here. And I really appreciate for always making time, and always ready to help and guiding in these forums, especially in the fight against IMR. We really appreciate that. And we are glad to hear that, moving forward, we are going to organize more workshops as well as maybe other training programs to impart more knowledge on the young people to continue to be active advocates against antimicrobial. Thank you so much, Lisa. Prof, over to you. You can say final word and advice. Something like that. Over to you, Prof. Professor, I hope you can hear me.
Yes, I can hear you.
Okay, thank you. Go on, please.
Just repeat for me, please. I had excused myself briefly.
Oh, okay. So just maybe you can tell us the final remarks. We are now ending the session.
Okay, so, final remarks. Thank you so much for putting this together. Been very delighted to be here to make a presentation, and I'll be around to make any other presentations in the future. And I also run a lab here in Mbarara, a molecular lab. So in case you need molecular laboratory services, PCR, real-time, ordinary PCR, you want to do ELISAs, you want to do Sanger sequencing, get in touch with me. We don't charge a lot of money. We are very price friendly. So we can help you to be able to find those genes. You are doing a research. We can also help you to really improve your methodology section. Many students have come to us with protocols that cannot really be done. So we can chat and see a way forward. Don't fail to do molecular work because they've told you it's very expensive. You can come, we will be able to help you. We have done it for other students.
Thank you so much, Prof. Okay, thank you so much, Prof. I think, one of the things that I actually limit research in Africa, maybe we can say on infectious diseases, is lack of facilities and maybe even the mentorship. Those are the challenges, I think, where the young upcoming scientists are facing, especially in the African continent. And I think with the kind of support that we have now, I think we are going at least to improve the capacity of the young people towards AMR research, discovery, and all that. Because I think once we have the support, the necessary support, and the willingness, there is nothing else remaining, only to find what you are looking for. And of course, you are going also to put more effort in the fight against this global health pandemic. And thank you so much, Prof. And thank you, Dr. Lisa. Yeah, we really appreciate your time. And we do hope that maybe coming forward for many sessions, we are going to work together here to ensure that I think we can impart more knowledge on AMR, especially also on the side of research. Thank you so much. And yeah, to all participants, I really appreciate
Jimmy?
it. Yes, Lisa?
Jimmy?
Yes, yes.
I'm seeing there is someone, Muzafaru, who has asked, we are in the era of AMR, but we gotten some information that chicken foods are mixed with ARVs. How can this problem be solved to reduce the resistance in future? I think this is to Prof, so that we don't leave anyone feeling they were not catered for. I've just seen that question.
Okay.
And I saw.
So that's very true. I think it was in the news here in Uganda
Last week,
that,
yes.
yeah, there are chickens that had ARVs. As we said, ARVs are used as growth promoters for feeds for chickens, especially broilers, to fatten them, and they grow very fast. So that's inappropriate use and actually leads to drug resistance. Because, yeah, once you get exposed to these low doses of drugs, you expose the virus to these low doses, it builds inherent resistance. And so the next person who picks up this resistant virus already now has resistance, so cannot use these ARVs. It's the same thing that happens with bacteria. Other people, for example, cough syrups, they go and pound anti-TBs and put them in the cough syrups. There was also a soda which I saw which had quinine in it. It was Novida which had quinine in it. So those are some of the really dangerous practices that we get involved in. Over to
Yes,
you.
Prof, and I think there has to be an holistic approach where even the authorities are involved, Ministry of Health, apart from health workers, also the Ministry of Health should be involved, agriculture, the vets. And I think that's why Jimmy had requested this talk about the transmission of these resistant genes in the environment. But to combat some of this, we may not say as students or as only health workers, but there has to be health education and also the involvement of authorities so that such practices can be curbed.
Yeah, there's something talk at length, but we don't have the time. It's called the One Health approach. That is the health of humans, animals, and environment. If we are to really curb AMR, we cannot avoid other sectors. We can't go solo. It can't work because what we do in health definitely affects the animals. If you look at the sewage that comes from the hospital, ends up in a sewer, ends up in a river, then animals are taking it, and those animals are going back to our homes. People are bathing in that water. You know, you go to the landfills where they drop, they burn all these drugs. Chemicals are left in there, and there are organisms in that soil. Somebody's walking there, he picks up an organism. So it's a cycle, and we need to be all involved. It can't be left to say Ministry of Health or health workers. It's a holistic approach. It's a whole society approach. And above it all, it's a behavioral approach. We need to bring social scientists on board and help us to understand behavior, because most of these are behavior issues. I mean, why do you have to put these drugs in chickens, which if you knew eventually are going to end up in the food chain, and you yourself may actually be affected. So it is very important for us to think like that. Olowe has raised the hand.
Yeah. Good afternoon.
Good afternoon.
Yeah, this is Professor Lowe from Nigeria.
Yes.
I love the last question on your presentation. There should be more awareness to the public, especially the poor rural settings where people are not aware of the danger of putting drugs into these chickens or animals. The One Health approach is a vital point that you have raised, and you mentioned they are in circles. But apart from that, it's more or less a behavioral pattern, our perspective to life, our awareness. People need to be told, beyond making money, profit from this investment, the danger it reposes back on the community. Those are areas where NSM should go on rampage awareness, massive awareness to the people, and effect to One Health approach. That's my own contribution.
Thank you so much, Professor. I mean, that's it. And it's, I mean, it's not pointing fingers to somebody, it is us. You know,
Yeah.
You know someone, you know a friend, you know a relative. Let's start word of mouth. You know, the people's revolution starts with people. You know, so we start and then others will catch up with us.
Yeah. Yeah.
Thank you
Sure.
very much, Jimmy. I have to run for some other activities.
Sure, Prof. Thank you so much, and we really appreciate your time and all the best.
Well,
Success
you're
ahead.
welcome.
Thank you so much. Yeah. Yeah.
Thank
I
you,
don't
Jimmy.
think-
Thank you, Prof. And thank you, everyone.
Okay.
Yeah, sure. Thank you. Thank you, Lisa, and all the best, success, and to everyone. I think, yeah, we're signing off for today's session. Let's meet coming next meeting. That's probably on September. We are going to announce that. Thank you so much. Until next time, have a wonderful time ahead and keep safe. Thank you.